So what about the screen? Can you see my screen? Can you see the presentation? Okay, excellent. Hopefully everyone can see, everyone can hear me. Hi guys, how are you? So I think I can start. The I will focus. So my conversation will be focused on the um, glial cells. Okay, glial cells. Do you know what glial cells are? Hopefully, and you know their main functions. But however, we will go again through these topics, and I will show you different images which will help you to understand their function. Okay, so you see here schematic image of central nervous system, gray matter, and you see here neurons. You know, here is indicated on this schematic image that this is part of white matter. Okay, but you know, we can use it for another part of brain too. But here you see that is the prevalence of myelinated axon. Okay, this is understandable. In the case of gray matter, of course, the prevalence goes to the unmyelinated axons. But you see here, this is to demonstrate how the oligodendroglial cells are working in the white matter. What you see here, you see neurons, look, neurons, myelinated neurons. You see here glial cells and their processes, which are attached to the surfaces of axons. And at the same time, you can see here other types of glial cells. For example, microglial cell, look, microglial cell, also um, astrocytes. And astrocytes are depicted here close to the blood capillary. It means that the blood-brain barrier is demonstrated here, of course, schematically. Also, you see here the small part of ventricle, lateral ventricle, and this lateral ventricle here is lined as all other ventricles and spinal cord canal too. They are aligned with the glial cells, which are called ependymal cells. So, what are the main functions of astrocytes? So they give rise to the blood-brain barrier. They regulate and monitor composition of interstitial fluid. But they provide very important real structural support and organization to the whole central nervous system. And they play very important role in the development, embryogenesis of the um, nervous system, because they are the main, they depict the main routes using which the neuroblasts have to move, neuroblasts have to migrate and reach the final destination places. What is this? You will see after 2 weeks, I think. After 2 weeks, according to our syllabus, we have there embryogenesis of nervous system. And then uh, we have also they replicate very intensively if it is needed, and they occupy spaces of dying neurons. Regarding the ependymal cells, they are also subtypes of glial cells, so they are actually cells which lines ventricles, line spinal cord canal, and they helps to produce the assist to produce the CSV, cerebrospinal fluid, and at the same time they provide its circulation, its movement from ventricles to the spinal cord canal and back. So regarding the microglial cells, they are phagocytic cells, real phagocytic cells, and they are able to move through the whole central nervous system, and they protect. Actually, they are a very powerful tool for protection of central nervous system because they are able to engulf different type of pathogens, infectious agents, and the um, harmful substances too. So we will start with the oligodendrocytes. Look, or astrocytes. Which one is first? I don't know here. Okay, astrocytes. But before, look please at the image. This is micrograph of gray matter. You can see here the neurons. You can see here glial cells. Look, glial cells. And whole this space is called neuropil. Yes, neuropil. So what is neuropil? Do you remember? Neuropil is extracellular matrix of nerve tissue, which is mainly constituted by the processes, neuronal processes, I mean axons, dendrites, and plus they also contain the processes of glial cells. So you see here this processes. Look, okay. This is hematoxylin eosin staining. Look, partial, but plus to this hematoxylin eosin staining is used also the gold chloride staining, because while this gold chloride staining, we can see not only cell bodies, but also their processes. Okay, so these are neural processes are here clearly visible. So these are glial cells. Oh, sorry, neurons. And you see here the dendrites. Look, dendrites, but of course not whole dendrite is visible. To reconstruct the three-dimensional image of the neuron, we have to use the reconstruction method, which very powerful softwares in the softwares. But here is most important thing when you want to reconstruct the or render the whole three-dimensional structure of glial cells or neurons. You have to be able to um, use all types of to keep all sections. And via this all sections, I mean, from when you section the material, tissue sample, you have to keep all sections. Only by these sections is possible to reconstruct the whole structure of the cells, or even this part of brain, or reconstruct the three-dimensional view of synapse, for example. So here you see the gray matter again, and you see the astrocytes. Look, this one, and these are oligodendrocytes. Sometimes it is not possible to identify, to differ from each other the oligodendrocytes and astrocytes, because it depends on the plane of section. But Sometimes it is possible, and usually oligodendrocytes are more ovally shaped or even round shaped, but astrocytes are elongated and they have such more ovally shaped. And from one side, their shape of nucleus is not uh, it is not symmetrical, you know. So the neurons are visible here also, and whole of this part of the tissue is neuropil. So what are the functions? Main functions of astrocytes: regulation of extracellular ionic content and around neurons and also around the synapses. This is the most important thing because they have to regulate amount of extracellular potassium and ions. Via this, the neurons will be able to function properly. And besides this physical support, they are also very, beside the physical, real physical support, they are also able to guide movement of neuroblasts, as I have already told you, during the embryogenesis. And they form blood-brain barriers. They form, yes, they form glial limiting membrane. When we will go to the meninges, you will see that they beneath the higher matter, they form glial limiting membrane, while the processes which reach the surface of the brain and gives rise to the very thin layer of glial processes, which is tightly attached to the surface of the brain. Is everything understandable what I say? Can you hear me? Can you see the screen? Is everything okay? Okay, regarding the micrographs, yes, they will be. And I will upload I upload micrographs today or tomorrow, okay? But hopefully you know that your first midterm is focused on epithelial tissue and skin. So, okay, now I go back to presentation. So, look, here is schematic image of astrocyte which participate in the information of blood-brain barrier. Look, the end tips of processes are attached to the surface of blood-brain barrier. This is astrocyte, and this astrocyte, look here, please. We have two types of astrocytes. Protoplasmic astrocyte, which has numerous processes, and all these processes undergo very intensive branching. And another one is gold chlorides, co-stained immunocytochemistry of astrocytes. And you see here the fibrous astrocyte. Look, this is fibrous astrocyte with the processes and with the and these processes are very well visible, clearly visible here due to the um, immunocyto chemistry. So I think I have once in last semester explained what type of method this is. Here are used the specific type of antibodies against the antigen in tissue sample. And such type of antigen, I mean specific protein for these, for fibrocytes, for astrocytes, are the glial fibrillar acidic protein, which is represented only in the glial cells. Okay, and we have here diagrammatically depicted direct immunocytochemistry and indirect immunocytochemistry. Here, only one population of antibodies are used together with appropriate labeling. Look, it might be, for example, peroxidase, or it might be immunofluorescent molecules. It does not matter. It depends on substance. And but in indirect one, we use two populations of antibodies. So unlabeled primary antibody which binds to the antigen in tissue sample, and then labeled secondary antibody. So as a result, this antigen is occupied by quite big amount of antibodies. So primary antibodies, secondary antibodies, and the secondary antibodies have this labeling. So after the appropriate processing, this antigen will be appeared. But when this big amount of secondary antibodies, which binds to the primary one, mm, amplifies the labeling of this antigen. That is why sometimes in some situation, this indirect immunocytochemistry is more useful, is more successful. Okay, so is everything clear with the immunocytochemistry? Okay, so look what we have here. We have here silver stained tissue sample. We have here capillary, and we have here astrocytes. And you see that processes of astrocytes are attached to the surfaces of capillaries. So this is brain blood barrier, capillary together with the astrocytes. Here is a TEM image of astrocyte, and look, this is the shape of nucleus via which we are able to identify them in the conventional staining. Look here or here, okay? Shape of nucleus. You see here, this is TEM image, and you see here part of cell body, neuron body, or perikaryon of the neuron. Look, this one here. And you see also the neuropil. Neuropil, this is the astrocyte, this one here. And usually the astrocytes, look, these dots, dark dots, they are intermediate filaments. The small part of cytoplasm is magnified here to demonstrate the intermediate filaments in the cytoplasm of astrocyte. So via this intermediate filament, we are able to identify astrocytes. Because, in the neocortex, or sometimes in other structures too, the small neurons, stellate neurons, and small fibrous astrocytes are very difficult to identify from each other. That is why we use this immunocytochemistry against the neurofilaments in the case of neurons, and um, against the intermediate filaments, GFAP filaments, or glial fibrillar acidic protein filaments in the astrocytes. Sometimes we use both of them. And when we receive, for example, the cells, because they have the same profiles in the tissue samples, they have sometimes the same sizes, besides the type of neuron, it depends also on the sectioning. So that is why when the neurofilaments appeared via the cytochemistry, immunocytochemistry, we can say that these cells are neurons. In another case, if the GFAP positive cells appear, it means that they are astrocytes. So what you see here in the neuropils, look, this differently shaped profiles. Look, even small ones, a little bit bigger here, um, you, or here, for example. Look. So dendrites, axons, but these axons are non-myelinated axons. Here, these profiles of axons are surrounded with a black thick line, and it means that these are myelinated axons. Look, the longitudinal section through the dendrite here. And here is the perikaryon membrane, or membrane which surround the cell body, neuronal cell body, and you see a rough endoplasmic reticulum, very well developed, together with a mitochondria. So mitochondria we can see in neuropil too, but in neuropil, mitochondria, of course, is represented in the axons and in the dendrites. But as you know, axons do not contain rough endoplasmic reticulum. Dendrites have, and also, but where mainly when they undergo branching and their diameter became smaller and smaller, this amount of rough endoplasmic reticulum or Nissl substance also goes down. So again, cross section through the myelinated axon, and but here on this here the magnification is huge, 57,500. You see the small part of cytoplasm of astrocyte, and you see here um, for example, smooth endoplasmic reticulum, intermediate filaments, mitochondria, and leaflets, two leaflets of nuclear membrane. What is here? Nuclear pore complex, of course. And here is chromatin. Look, attached to the inner surface of inner leaflet of nuclear membrane. Okay, so this magnification here is 12,000, here is 57,500. That is why you see here only small part of cytoplasm. So this is, again, brain section stained by immunohistochemistry against this GFAP. And you see here the astrocytes. Not other type of cells are stained here, only astrocytes. Look, only astrocytes. This is highly specific type of staining. Okay, another type of glial cells are oligodendrocytes. And look, oligodendrocytes, cells which myelinate the axons. And please look at this schematic image. They send their processes to the surfaces of the axons and wrap the small part of the axon into the myelin. One glial cell, one oligodendrocyte, is able to myelinate several axons. And they myelinate several parts of axons. Okay? And this amount of axons might reach from 30 up to 80 exons. So look, this is white matter, and uh, you see here astrocytes, and sorry, this is myelinated axon. Look, these are such type of profiles, or this one, for example, they belong to myelinated axon. And also nucleus of oligodendrocytes are clearly visible. But the profile of axon is usually surrounded with a light hallmark of light rim of myelin, which during the tissue processing is dissolved in the fixatives and different type of substances which we need for clearance, for example, of samples. And that is why because lipids easy are dissolved in these substances, and you know that the main part of myelin is constituted by the lipids. So here is again schematic image how the one oligodendrocyte myelinate the axon, and you see here these processes and the um, layers of myelin which covers the surfaces of these myelin layers, this myelin sheath. They are unrolled here to demonstrate how it happens. Okay. So, in the case of astrocytes, the axon, in the case of Schwann cells, the axons are embedded in the cytoplasm of Schwann cells. But here, in the case of oligodendrocytes, they send the processes, and via these processes, they wrap the axons. Is it understandable what I say? Is understandable how they myelinate. I mean, difference between the myelination via the oligodendrocyte and myelination via the astrocyte, via the Schwann cell. So axon is embedded in the Schwann cell cytoplasm, cell itself, and undergoes myelination there. I will show you the micrographs a little bit later. But in the case of oligodendrocytes, they send the processes, look, and myelinate, wrap the exons. Okay. So here is special type of staining using which we can um, reveal myelin. Look, this blue stain are myelins. This is cross section through the myelinate or transverse section through the myelinated axons. Here is longitudinal section through the myelinated axons. The same is here. So, and here the oligodendrocytes, their nuclei are clearly visible. So this is special type of staining, which is called solochrome cyanin staining, via which we are able to stain the myelin, and also the profiles of oligodendroglial cells are also clearly visible. Another oligodendrocyte, but now it is demonstrated via the TEM image, and here the magnification is 13,000, but however everything is clearly visible because the zooming is too high. You see here the cell perikaryon, look, part of perikaryon, look, this one here. So the part of nucleus, sorry, part of nucleus is here part of cytoplasm. And the cytoplasm contains a lot of mitochondria, lot of free polysomes, and also the rough endoplasmic reticulum, and Golgi complex is clearly visible, a lot of polysomes. So the Nisl substance is very well developed here. Active neuron, because nucleus is also euchromatically stained. And whole this part, look, all this part represents the neuropil. And what can you see in the neuropil? When you look at the PEM images of the neuropil, it is much more easy to understand what are the main components of neuropils. So glial cells themselves, okay? Look, this is cell body with a well-developed Golgi complex, plus the rough endoplasmic reticulum, nucleus, which is euchromatic mainly, and here you see myelinated exons, unmyelinated exons too. Look, unmyelinated exons, and a lot of synaptic connections. Synapse 1, synapse 2, synapse 3, 4 here, 5, 6, etc. So look how we can identify synaptic connections. We can identify via the active zones. Look, these dark lines, okay? Dark lines. On the one side of this active zone, we have axon, which is presynapse and contains a lot of vesicles, vesicles filled with the transmitter. And another part, another on another side of the active zone, you can see the dendrite. It might be dendritic spine, it might be dendritic shaft, it might be soma, part of perikaryon of the neuron, because we have axosomatic synapses too. Or and usually, look here, for example, you see here the synapse, and because active zone is represented here. Active zone, what are the components of active zone? What they contain? Presynaptic membrane, postsynaptic membrane, and synaptic cleft. And on the one side of this active zone, we see here vesicles and mitochondria, and another part, on another side of this active zone, we have dendritic shaft or dendritic spine. So this is postsynaptic portion, presynaptic and postsynaptic, and active zone with a presynaptic membrane, postsynaptic membrane, and synaptic cleft between them. Is it understandable? Maybe you have questions regarding the uh, structure of synapses. Everything is clear. Can you hear me? Can you see my screen? Okay. Yes, for example, this profile here, they are cross-sectional. Cross-sectional. This, for example, profile of myelinated axons, they are more obliquely sectioned. That is why it is elongated profile, and but this round shaped one, this is definitely more roundly shaped. This is definitely transverse section. This is also transverse section, but it is more oblique. Okay, but here, have we here longitudinal sections of dendrites or axons? Look, look, this one, small fragment of dendrite in longitudinal profile is visible here. Okay, so another type of glial cells, which is microglia. Microglia, again, look here, again, are used monoclonal antibodies against the antigen which is represented in the microglia. So when we stain via the routine staining, for example, hematoxylin eosin, you can see only the dark nucleus. That's all. But if you use this immunocytochemical staining, you can see the processes of this microglial cells. And you know what is characteristic for these microglial cells? They are cells which are represented in the nervous system solely. So they are not interconnected. They have to be able to move from one place to another place. And they also are derived from the monocyte precursor cells. And have they are less numerous than astrocytes, but the amount is approximately the same as amount of neurons, and they are um, if you they are able to scan with their processes the neuropil. They monitor content of the neuropil. They remove damaged synapses. They remove cellular debris, and this is very important in the case of injured brain or spinal cord tissue regeneration. This is a very important moment when the debris, when this damaged cells and part of cells are uh, removed from the tissue on time. So this is not happened in the case of brain, but in the case of spinal cord, it is peripheral nervous system. It is possible. In the spinal cord and in the brain, not so well as in the case of, for example, the peripheral nervous system. We have talked about this at the end of lecture of nervous system. Okay, but please remember that in the case of peripheral nervous system, the macrophages, microglial macrophages, are able to remove everything very on time, but not in the case of brain and spinal cord. Microglia here is not able to remove it on time. That is why the one of the factors which interrupt the healing, interrupt the repair of the nerve tissue is this one, together with, of course, glial scar. So, what else? They are able to remove different type of pathogens. They are able to secrete a number of cytokines, which are very intensively involved in the immune regulation. And when they became activated by products of cell damage, for example, or by invading microorganisms, they undergo several changes. First of all, they retract their processes, and immediately they start the phagocytosis, and they engulf the damage or danger related materials. And then when they process these materials, they are able to present the antigen on their surfaces. Okay, again, microglia stained here. Look, this. And round shaped nuclei are mainly oligodendrocyte. This is quite small magnification. A little bit bigger, you see magnification, you see the microglial cells and oligodendrocytes too. Okay, ependymal cells, which are epithelial-like cells, and they line the surface, inner surface of ventricles of a spinal cord canal too. And they have cilia on their surfaces and microvilli too. So via this microvilli, they are able to assist the formation and maintain the content of cerebrospinal fluid. But via the cilia, they are able to move to provide circulation of this cerebrospinal fluid. They have no basal lamina. Despite they are epithelial cells, they are derived from ectoderm. Ectoderm, they have no basal lamina. And their ends are tapered and they extend into the deeper parts of brain tissue. And they might be attached, for example, to the some processes. And uh, look, these are ependymal cells with a nucleus, with a free surface directed to the lumen of or ventricular space. This is ventricle. These are ependymal cells with their nucleus, with their processes, which goes into deeper layers of neuropil. And here is the system, whole system of the ventricles, ventricular system. So you see here lateral ventricles. Look, whole this structure is lateral ventricle. Okay, first one, second one. Then in the middle, we have two lateral ventricles. In the middle, we have here 1/3 ventricle, and this first and second lateral ventricles are connected with the third ventricle with the foramen, interventricular foramen, so they uh, connect the lateral ventricles to each other, plus they connect them to the third ventricle. Then the whole third ventricle, look, this is third ventricle, is connected to the fourth ventricle. This connection is mesencephalic aqueduct here. And the fourth ventricle here has lateral aperture and median aperture. It means real holes via which this ventricular system is connected with the space surrounding the whole brain and spinal cord. So this is spinal cord canal. The inner surfaces of these ventricles are covered with the ependymal cells. Plus, in these ventricles, for example, lateral ventricles, we have uh, also the type of tissue which is the main producer of um, cerebrospinal fluid. And this type of tissue contains epithelium, contains loose connective tissue because it is very well, that is why it is very well vascularized. And actually, the cerebrospinal fluid is derived from the blood. Look, this is transverse section through the spinal cord canal, lumen of spinal cord canal, tissue sample stained with hematoxylin eosine, and you see here cells, ependymal cells, which lines the surface of spinal cord canal. And this is surrounded with gray matter. Okay, another type of glia is peripheral neuroglia, which contains Schwann cells and or neurolimocytes. They are called also neurolimocytes. We have satellite cells there and variety of other cells which are associated with the specific organs. For example, glial enteric glia or Muller glia, which is in the Muller cells in the eye. So in the retina, the satellite cells are mainly represented in the root ganglion, dorsal root ganglia. They are, look, shaped ovally, quite big sized cells, and they are surrounded with a monolayer of satellite cells, which actually are not satellite cells. They are neurolemocytes or Schwann cells. Okay. So, you know what is the main feature of this dorsal root ganglia? No synaptic connections are observable in the uh, dorsal root ganglia. That is one of the main difference between the dorsal root ganglia and ganglia of autonomic nervous system, for example. You see here the motor neuron, which gives rise to the axon, and you know that these axons are very long, and part of the axon is included in the gray matter. This is gray matter here, schematic image, but I think it is very good image, very understandable. And here you can see the axons, the axon hillock, and then initial segment of the axon, and then whole axon which undergoes myelination. How do you think? Which type are they? How they undergo myelination? So here is indicated that Schwann cells. This is the Schwann cell. This nucleus belongs to the Schwann cells. So Schwann cells here myelinated the whole axon. Am I right? Am I right? Are they able to myelinate the whole axon, Schwann cells? For example, axons of motor neurons. Of course not, because no. So, Mustafa, they are not able to be produced from the ectoderm. We have ectoderm during the embryogenesis, but and regarding the proliferation of ependymal cells, they are not able to actually proliferate, you know. But this is very good questions. I will check. Maybe we have uh, last data according which this might be changed. I will see. Okay. So, what about the node of Ranvier? Node. You are answered my question. So, Schwann cells are not able to myelinate the whole length of motor axon. Of course not. And do you know why? if you go to this image, I mean this schematic image again, and I will show then micrographs too. So you see here, look, part of the axon which is incorporated in the gray matter. So here oligodendrocytes are working very intensively, but immediately when this axon left the spinal cord and became the peripheral nerve, in this case peripheral axon, in this case Schwann cells to start to myelinate these axons. Is it understandable? Is it? Okay. So um, these are alive cells, alive swan cells in the tissue culture, and they are properly stained via the immunostaining. That is why they are clearly visible, and they looks like very nice here. So, but in the tissue samples, this was tissue culture. Here is tissue sample and fixed, stained, and sectioned and stained. And you see here nodes of Ranvier. Here, look, nodes of Ranvier and nucleus of Schwann cells. Okay. So the Schwann cells they regulate very intensively the content of the myelin. They give rise to the nodes of Ranvier, and they are involved in the regulation of the ionic content, ionic microenvironment around these nodes. So the interaction of Schwann cells to the axon is very important because they stabilize the structure of axonal membranes, and at the same time they provide clustering, they provide grouping of sodium channels at the nodes. um, these are satellite cells and the cells of neurons of dorsal root ganglia, but these are satellite cells. Do you know what this dark brown staining means? That these neurons contain lipofuscin granules. So what was lipofuscin? Lipofuscin was, do you remember lipofuscin? Residual bodies and concentration of lipofuscin granules. So lysosomes which contains not more digestible material plus lipofuscin granules. Okay, guys, 2 minutes more and then you will be free because I think you have another tutorial. Yes. So here you have immunofluorescent staining again of satellite cells. Look, after 1 hour, you have the histology. Oh, sorry, anatomy at 3. Okay, in this case, can I prolong? Can I talk with you about the neurons too today? Is it possible? In this case, I will do this, okay? Well, thank you. So I will finish glial cells and we'll go back to the neurons. So what we have here, we have this cells stained immunocytochemically. Immunofluorescent staining is, but you know what does it mean, immunofluorescent staining? It means that the antibodies are labeled with a immunofluorescent dye, and this immunofluorescent dye is able to emit the appropriate light at the appropriate when if we use appropriate filters in the microscopes. So here is rhodamine red labeled antibodies. Okay. So another, okay, look, this is 10 image of fibrous astrocyte. And you see here, look, this is cell body. Look, you see here cell cytoplasm and protrusion of this cytoplasm. It forms a small part of the um, process of astrocyte. And you see here a lot of bundles of filaments. Look here too. So what type of filaments they are? They are GFAP, glial fibrillar acidic proteins. So fibrous astrocyte surrounded with a neuropil. And what you have here in the neuropil, look, this is process number 1 of astrocyte, process number 2 of astrocyte. This is cytoplasm of astrocyte body, very active cells, almost wholly eukaryotic. And you see here, look, the transverse sections through the myelinated axons and through the unmyelinated exons too. Can you see here the active zones? Can you see here the synapses? Look, here is visible. It is too small magnification for, um, or here. Look, this is synapse 2, active zone, presynaptic axon, and postsynaptic one. Look, I'm talking about the, oh, I have lost it. So it was somewhere here. Look here, for example, pre-synapsis and post-synapsis. Pre-synapsis is always more dark stained than post-synapsis because pre-synapsis contains usually a lot of vesicles, and these vesicles are filled with the transmitters. Okay, so, and you see these filaments. This is process of the astrocyte. Another process, look, another one is two here. Look, this one, two. Okay, what we have here. Here we have oligodendrocyte. So this is white matter of central nervous system. And you can see here anterior commissure. Do you know what anterior commissure is? This is part of white matter which connect the hemispheres to each other. So this is one of the oligodendrocyte, nucleus, cell cytoplasm, and a lot of, almost all exons which surround this profile of oligodendrocytes are myelinated. Which of them is myelinated, where this exon is a little bit difficult to identify, but you know that the quite big part from 30 up to 50, 50, profiles of the exons are represent the exons which are myelinated by this cell, by this cell, okay? So look, profiles, profiles, profiles of different sizes, and they are myelinated. Another part of This is much more smaller magnification, again, anterior commissure, and you see here astrocyte. Oh, look, astrocyte is here, oligodendrocyte is a more round shaped nucleus. And plus the microglial cells are visible here. Look, and all these cells are surrounded with myelinated axons. You have to remember that in the white matter, we can identify neurons too, but the amount, of course, is not so big as in the case of other parts of different parts of gray matter. So what you see here, this is the microglial cell. Look, microglial cell. Do you remember microglial cell? Roughly, this is the macrophage of central nervous system. A little bit different origin. Macrophages of connective tissue, for example, they are derived from the monocytes, and the microglial cells, they are derived from the uh, blood cell precursors of monocytes. So nucleus and engulfed material. Look, they are arranged solely in the tissue. They are not interconnected. They are able to move. They are able to engulf the debris of the cells, and plus they are able to monitor the content of neuropil. Okay, so this is more zooming, and also look here, these different profiles. It will be maybe it will be interesting for you. Look, we have here this microglial cell in the middle, in the center, and this microglial cell is surrounded with a neuronal cell body and dendrites, and they are stained blue. Look, this is neuron, of course, this one here, and here are the dendrites. Look, longitudinal section through the dendrite, cross section through the dendrite, oblique. No, you know what this is? This is the typical exospiny synapse. Look, this is axon, this one, and that one is dendrite. And this protrusion is small spine here. And other spines are represented in the gray color too. Look, this very, very thin, very, very small. But this one is spine too. Look here also, because we have here shaft and we have here this small protrusion. Or it might be the shaft in a little bit different profile of sectioning. Hmm. However, spines are here. Look, but I think that this is the shaft and this is the small protrusion which gives rise to the spine. Okay, so what you see here are also astrocytic processes, and they are stained yellow. Look, look, huge amount of astrocytic processes, and they fill the neuropil. So the content of neuropil is very well visible here. We see here synapses. We see here myelinated axons. We see here unmyelinated axons 2 and axon terminals. We see the dendrites. So this image gives really very well understanding about the content of neuropil. Okay. So, is everything clear? I hope. Look, this is normal white matter, normal white matter. And here is schematically depicted how the one glial cell is able, oligodendroglia is, or oligodendrocyte is able to myelinate several, in this case, 3 exons. But this is example of oligodendroglioma, and this is very aggressive type of tumor. And if you compare amount of glial cells, look, these dark dots are nucleus of glial dots of glial cells. You see how the how it differs from each other. So these are blood vessels here, blood capillaries. So again, myelinated axon, again, oligodendrocyte. Look, myelinated axons, oligodendrocytes. Look, the nucleus looks like a astrocytotic nucleus. That is why in such cases, the immunocytochemistry is very important approach. So now look please at this image. You have here cell nucleus, cell cytoplasm, okay, and myelinated axon, which is embedded in the cytoplasm, which is surrounded with a cell body cytoplasm, not processes, no, cell body cytoplasm, okay? And this is myelinated axon. So immediately you have to say that this is swan cell. Swan cell. Another swan cell. Look here with a myelinated axon in the cytoplasm. Small part of cytoplasm is clearly visible here, but you can't see here the nucleus. No. Um, Look here, please. Here are again swan cells and exons in their cytoplasm, but these are unmyelinated exons. And usually size of unmyelinated exons is much more smaller than myelinated one, and uh, you can see in the peripheral nerves both of them. I mean, myelinated exons and unmyelinated exons too. So they are again surrounded with swan cells, and small cells have to accomplish their function here too. But regarding the myelination, these exons are not myelinated. Is everything clear? Is everything understandable regarding the myelinated exons? Mustafa, the transmitters should be amount of transmitters should be regulated in the synaptic cleft and around the synapse. And here the key players are not only astrocytes but also endocytosis, which is mediated by especially receptor mediated endocytosis, via which the presynaptic part of synapse is able to uh, absorb this extra amount of transmitter. So via this, the very precise, very proper transmission is maintained in the synapses. Okay, is it understandable? So not only astrocytes, but also the presynaptic ends too. Okay, regarding the swan cells, is everything clear? So, do you remember this image? Oligodendrocyte with the embedded, with the, sorry, with the, is surrounded with the profiles of myelinated exons, and quite big amount of cytoplasm is clear of exons from exons. In the case of oligodendrocytes, look, I'm talking about this one. Look, this is cytoplasm of the um, oligodendrocytes, and no exons are in the cytoplasm. But you see here the processes of oligodendrocytes which surround these blocks of myelinated exons. Okay, but in the case of swan cells, you see the cell profile, I mean profile of the myelinated exon, which is surrounded with the cell cytoplasm, or even nucleus is clearly visible here. And these are unmyelinated exons, okay? Unmyelinated exons, again surrounded with the cytoplasm of swan cells. But swan cells here, they do not produce the myelin, okay? So, um, I want to go to another. Have you have maybe you regarding the neurons? Maybe you have questions. I mean, uh, their structure. Hopefully, you know about the Parkinson disease. Yes. So Parkinson disease, look, which is characterized by tremor, reduced activity of facial muscles, and loss of balance, and also stiffness of posture. So the cell bodies, which lies within the substantia nigra, look here, they step by step they undergo degeneration. So here is the normal substantia nigra, and you see the shape of this nucleus looks, especially this part, it looks like as a swallowtail. That is why the swallow is presented here. And look, one part, another part, one part, another part. So this is healthy individual. In the case of Parkinson disease, this swallowtail sign is absent. Okay, so mm, this is Alzheimer disease brain, and in the case, of course, this is schematic image, and here you can identify this amyloid plaques and also the neurofibrillar tangles in the cell cytoplasm. So these flaps are identifiable outside the cell in the tissue, and the tangles, these neurofibrillary tangles, are identifiable in the cell cytoplasm. So these neurofibrillary tangles, they actually accumulate in the cell cytoplasm, and they are associated with the microtubules. You can see them also in the axon hillock. Regarding the plaques, they are aggregates of protein, beta amyloid protein, which is formed outside the cell cytoplasm. So this is real tissue samples of plaques. Look, plaques. And here you can see the tangles in the neuron. So Alzheimer disease. Um, so what are these? Spines. Yes. This is silver stained neuron. Yes. Silver stained neuron. And here is demonstrated the motility, flexibility, or dynamic structure of the dendritic spine. So this is confocal scanning microscopy, which and during several days these images were captured. And you see here the blue sorry, blue arrowheads, which indicates the stable spines, population of the spines, which is represented during this whole imaging process. And you can see also the green, the spines indicated with the green color, green arrowhead. So they are newly formed spines. Look, this one, for example. And this one then became stable here, stable here, stable here. But another one, for example, this one, it was not here, definitely not here on the first day of capturing, and then the small protrusions is derived, then the spine is finally formed, but after 4 days it is fully disappear. So, and this features of the spines is very important because they, by this way, they provide highly plastic nature of brain of connections in the brain, in the central nervous system. They are able to establish new circuits, neural circuits, I mean microcircuits or even macro circuits, and they are also able to reorganize these circuits. Is it understandable? Is understandable regarding the spines. Okay, what I want to show you is also the, so this is exon hillock, and here is initial segment. So in the initial segment, actually, the look, so this is the cell body, dendrites, and here is the axon hillock. Look, this part, and all other part is initial segment. So in the initial segment finally is triggered an initiated nerve impulse. That is why very often you can identify exoexonic synapses in this part, because these exoexonic synapses are very often inhibitory too. And by this way, they modulate activity of this neuron. Okay, look, then right. Okay, what we have here or here. I want to focus again on the um, neuropil and synapse. Okay, so what we have here cell body, axon hillock, initial segment. What we have in axon hillock, a lot of microtubules are clearly visible, together with the microtubules, of course, intermediate filaments, which are neurofilaments. Can you see here active zones or synapses? Look, one, two, three, four, five, six. Here, this one. A lot of synapses, or here, or here, or here. So what is the active zone? Hopefully you know. This is pre-synaptic membrane, postsynaptic membrane, and synaptic cleft. And on the one side of this is whole synapse. Look, this one. And here is active zone. On the one side of active zone here, you can see uh, this is axon, yes, because it contains transmitter vesicles, and this is dendrite. Quite big profile of dendrite. Look here, this one, that one, this one, that one, here too, and this one too. So on this image, you see this is much more higher magnification. Again, neuropil is visible here without cell body, and you see the axon initial segment, which is magnified. It contains mitochondria, microtubules, neurofilaments, look this one, or here that one, and cisterns of smooth endoplasmic reticulum, but no polyribosomes of methyl substance. In the dendrites, you can identify polyribosomes, but in axons, no, never. So, synapse, this one or that one, synapse, or here, and look, please, this is exosynaptic, exodendritic synapse, and here this is small protrusion which gives rise to the spine, but no uh, synapse is identifiable here. Looks the same as here, but we can see here the small part of neuron, perikaryon of the neuron, nucleus, and methyl substance. Look, very well developed, but no methyl substance in more farther part of uh, axon hillock. In initial parts, you can see small amount of free polyribosomes, but farther, no, of course not. Okay, look, this is structure of the synapse. You see here schematic image of pre-synapse, and this is post-synapse, and you see here the vesicles, mitochondria, and microtubules too. Via these microtubules, the vesicles reach the presynaptic part. But you know, when they reach this part and when they became empty, then later they are refilled with a transmitter. We have to here two type of mechanism of synaptic transmission. So one is both of them are uh, very effective, and one during the one the synaptic vesicle is fully fused with the membrane and release its component, its transmitter outside. In another case, it might be fused via the porosome, this is special type of structure, in the presynaptic membrane, release part of transmitter, or fully release the transmitter, and then it undergoes refilling. The vesicles which are fully fused, they have to be formed de novo again. So from ergonomical point of view, the porosome are very convenient for the cell because new here the energy goes to the refilling process, not re establishing or recreation of the synaptic vesicles. So here you see this presynaptic part, and with the mitochondria, with um, vesicles, and myelinated part of axon is also visible. Here is cleft, look, synaptic cleft. This is dendrite here, and mitochondria. Here we have, look, this is post synapsis, but however, we have here vesicles. That is why it is another terminal. So axonic contact from this side here, and this one is exodendritic. But here we have axonic contact. Synapse. Another synapse. Look. Again, this is part of neuropil, of course. And you see here the presynapse with the axon and vesicles, mitochondria, and this is postsynaptic part. And here is the schematic image of porosome complex. Look, this is porosome. So two types of neurotransmission. One when the vesicles fuse fully with the presynaptic membrane and release their content, and then they have to be established again. And another one via the porosome complex. So they look, this is um, cytoplasm, cytosol here. This is outside surface of the cell membrane. Look, peripheral proteins are visible here, but and in the thickness of the membrane, the porosomal complex, this one is embedded. So the opening of porosomal complex is visible here, and the base, where usually the synaptic vesicles fuse, is represented there. So we have here very fine mechanism via which the synaptic vesicle membrane is able to fuse with the base of porosome complex, and this is called via the T and V sneer membranes. So vesicle associated membranes and target associated membranes. So their fusion provide this binding of synaptic vesicle to the porosome complex. And in the middle it has plaque, and via this movement of the plaque, porosomal complex might be open or closed. So when it goes up, in this case the lumen of the porosomal complex became open and the transmitter is released outside. Is it understandable? Is it understandable? Okay. So guys, I think I have told you everything what I want to stress, but however, maybe you have questions. Look, this is TEM image, and you see here synaptic vesicle and um, porosomal complex. This is made, and this process, this tissue, and these samples are, and this one also are received in the lab where I am working too. I'm very proud because this is really very interesting work, very interesting work. And here you can see, look, the different type of neurons, neurons of cerebellar cortex. About this, we'll have talk next week. I mean, type of neurons in the center, how the central nervous system is structured, and how the peripheral nervous system is structured. Well, you can see here um, the neurons which are stained with the silver staining. That is why their processes are clearly visible. But regarding this one, for example, this one is hematoxylin eosin. Sorry, no, this is toluidine blue staining. Or another way, it might be stained. It is called also Nissl staining, and you can see this dark stained cytoplasm, which contains very well developed rough endoplasmic reticulum and free polysomes. Okay, and what we see here, do you remember what type of tissue is this? What type of tissue is this? Yes, exactly. Loose areolar connective tissue, this one. And uh, what type of cells are these cells? For example, this one, or that one, or this one. What type of cells they are? Exactly. Thank you, Mustafa. And here you can see also, thank you, Esther. You can see here the other type of cells too, which are, look, this one, or that one, or that one, this one. Fibrocytes, inactive fibroblast actually, or less active. So uh, why I put these slides, I mean this micrograph together, to demonstrate how the extracellular matrix differs in these tissues from each other. So this is extracellular matrix of loose areolar connective tissue with a ground, clearly visible ground substance, very well visible collagen fibers. And the same is here on the TAM. You see the small part of the fibroblast and a small part of fibroblast and extracellular matrix with the collagen fibrils with the elastin fibers here, and collagen fibrils are longitudinally sectioned, these are cross section. But what is in the case of neural nerve tissue? We have neuropil instead this type of extracellular matrix. Of course, ground substance is here, small amount of ground substance, of course, uh, but actually the whole neuropil is constituted by the processes, cellular processes, glial cells, and synaptic connections. Look here, typical neuropil with the synaptic connections. These dark lines are active zones, and with the pre and post synaptic, and with the profiles of different type of processes, dendrites, axons, glial processes, longitudinal section through the axon, dendrite. So sorry, dendrite. Okay, guys, I'm done with my tutorial. Maybe you have a question. So you can use your break time before the next lecture or tutorial will be started. If something will be not clear, you have questions will be questions exist, please feel free, write me. Okay. Okay, excellent. In this case, if you have no questions, I wish you health first of all, and meet you next week. Okay. Bye guys.